Hi everyone, welcome to my channel. Today we're going to learn the second topic of Theme A, Unity and Diversity from Organization 1 Molecules, Nucleic Acids. Before diving into our lesson, let's see what are the learning outcomes. These learning outcomes are very important depending on your level, either your standard level and HL level. You should learn properly because contents vary depending on your level. So look through this content before starting. Now, in the new syllabus, we have the guiding questions at the beginning of the lessons. So here, related to nucleic acids, we have two guiding questions. How does the structure of nucleic acids allow hereditary information to be stored? And how does the structure of a DNA facilitate accurate replication? At the end of the lesson, you will be able to answer these two questions. The organisms alive on Earth today have a very long history and a very long family tree. Living things do not just appear, rather they descended from previous generation, and this is based on the genetics. The information that is being passed from one generation to the next is the form of the DNA. So DNA is the genetic material not for the all but most of the living organisms, because we have the another type of nucleic acid which is called RNA or ribonucleic acids that for example some viruses use RNA as their genetic material, such as the coronavirus, which is very famous, and the HIV. So when we were talking about the genetic material, so try to understand that it's a store of information. If copied, it can be passed from one cell to another cell, also from parent to offspring. Because genetic material is inherited, it is sometimes called hereditary information. So most of the organisms use DNA as the genetic material. And the full name for the DNA is deoxyribonucleic acids, and the other type is called ribonucleic acids or the RNA. Now, components of a nucleotide. So, what is the nucleotide? Both DNA and RNA are polymers of nucleotides. So, what does it mean? It means that DNA and RNA are composed of many nucleotides. And each nucleotide consists of three parts: a pentose sugar, which is in the purple color, which has 5 carbon atoms, and the pentose sugar varies depending on the either it's a DNA or RNA, a phosphate group, which is the acidic and negatively charged, and the base that contains nitrogen and has either one or two rings of atoms in its structure. And covalent bonds occur at specific locations between the pentose sugar, a nitrogenous base, and the phosphate group. Okay, so the sugar phosphate bonding and the sugar phosphate backbone of a DNA and RNA. As we said, covalent bonds are formed between the phosphate of a nucleotide and the pentose sugar of the next nucleotide. The nucleotides are always added to the growing polypeptide in the same way the phosphate of the nucleotide being added is linked by a covalent bond to the pentose sugar of the previous nucleotide. This chain forms a strong sugar phosphate backbone in DNA and RNA molecules that helps to conserve the sequence of bases. Nitrogenous base within nucleic acids. In total, there are 5 possible nitrogenous bases in RNA and DNA. 4 are found within RNA and 4 are found in DNA. Only one of the bases differ in 2 types of polymers, so that it may look like some of the nucleotides found in RNA and DNA are identical. For example, both polymers contain adenine, cytosine, and guanine. The only different nitrogenous base is thymine for the DNA and the uracil for the RNA. So instead of thymine in the RNA, there is a uracil, and instead of the uracil in DNA, there is a thymine. The structure of RNA. RNA is formed when nucleotides become bonded together in a very specific sequence, And the nucleotides are joined together by a condensation reaction between pentose sugar of one nucleotide and the phosphate group of the next nucleotide. And the reaction releases a water molecule. If an RNA molecule contains, for example, 322 nucleotides, the 321 molecules of water would have been produced during its synthesis, and it would have required 321 condensation reactions to form. So remember that RNA is composed of a single chain, while the DNA has two chains of these strands of a nucleotide. The structure of DNA. The DNA has a double strands, and you can imagine as a ladder that are made up of the phosphate and deoxyribose sugars. The rungs of the ladder are made up of the nitrogenous bases, and the bases are always adenine bonded to thymine and cytosine bonded to guanine. And there are no exceptions to this in DNA, and these base pairings are known as the complementary base pairs because the two are said to be anti-parallel. The distinguishing between DNA and the RNA, so including the number of the strands present, the type of nitrogenous base, and the type of pentose sugar are different in DNA and RNA. So the DNA has a double-stranded molecule, whereas RNA is single-stranded molecule. The DNA has the deoxyribose pentose sugar, whereas RNA has the ribose sugar. The DNA has a thymine as a nitrogenous base, whereas RNA, instead of thymine, there is a uracil. And DNA has a double helix shape, whereas RNA can be in a different shapes. DNA is a permanent genetic code for cell and organisms, but the RNA is not the permanent genetic code, and there's an exception. These are the only viruses. The importance of the complementary base pairing. So what is that? Is that the complementary base pairing is important in DNA replication. In DNA, adenine can only pair with a thymine, and cytosine can only pair with a guanine. And this is the complementary base pairing, and it allows the exact copy of a DNA molecule to be made in a process called the replication. And this is very important, complementary base pairing. Diversity of possible DNA-based sequences and the limitless capacity of DNA for storing information. DNA stores genetic information in its sequence of nitrogenous bases. Every 3 bases represents a meaningful piece of information called a triplet, or more specifically, a triplet codon. Many triplets within DNA code for one of the 20 amino acids, which are very important. And there are 4 different DNA nucleotides that can be arranged as sequence triplets. So what are the odds of DNA containing any one triplet in any one genetic location? So that the consider the odds of having GGG in one triplet area of a DNA. It was by a random chance that the odds would be so there are going to be 64 codons for that. And the diversity of possible DNA-based sequences means that DNA has limitless capacity for storing information, and with a diversity by any length of DNA molecule and any base sequence possible. In human cell, the DNA held in a nucleus measures about 20 meters in total length, and these lengths contain 3.2 gigabyte of the data, a phenomenal quantity of genetic code. And within this DNA, it's estimated that humans have 20,000 to 25,000 protein coding genes, and this is a really great economy for storing the data. There are 64 codons because each base in the codon can be any of 4: the adenine, thymine, cytosine, guanine, and there are the many combinations, the 64 combinations, and each 64 codons has a meaning. Most codons specify one particular amino acid, and 1 codon signals the protein synthesis should start. 3 codons signals the protein synthesis should stop. And all living organisms and all viruses use the same genetic code to represent the sort of the genetic language. Many different versions of genetic code could be devised and they would probably function perfectly well, but all life forms use essentially the same version. For this reason, it's called the universal genetic code because all life forms use genetic material. Conservation of the genetic code across all life forms and as evidence of universal common ancestry. What is that? The organisms different from each other, right? So the answer is that the why organisms are different from each other is that the DNA based sequences are different, even though the code to read the sequence is the same. Your best friend, although not directly related to you, is related to you by evolution. The two of you share more than 99% of the same gene sequences. If it was 100%, you would not be the unique and different people you are. Why the genetic code remained unchanged? The answer to this question lies in the process of evolution. And for over 3.5 billion years, that the only most minor variations in the genetic code have arisen. And the over many generations changed in the sequence of bases in the genome and therefore in the mRNA and the order of the amino acids they assemble can occur due to the mutations. Many sequences, both in areas which code for protein, which we call coding sequences, and that do not, which is known as non-coding sequences, persist unchanged. However, with only minor modifications over the many generations. Even if the base sequences in coding areas of DNA change, the sequence of amino acid may not or may change, or the RNA, mRNA codes and mutations in a coding sequence do not necessarily affect the amino acid sequence of its protein product. So the most highly conserved genes are those that can be found in all organisms. These include proteins required for transcription and translation and those found in ribosomes. The fact that such genes exist indicate that all life is interlinked with the universal ancestry for all life on Earth. Directionality of DNA and the RNA. The 5' to 3' designation refers to the fifth and the third carbon atoms in the ribose and the deoxyribose sugar. In a DNA molecule, there are two strands that run anti-parallel to each other. This means that if you compare the sides of DNA, one strand will run from 5' to 3' and the other will run from 3' to 5'. And this does not mean the direction in which they were synthesized is different. Both strands of DNA are synthesized starting with a 5' nucleotide and working towards the 3' end. So the information in the DNA lies in the sequence of the bases: cytosine, guanine, adenine, and thymine. This sequence dictates the order in which specific amino acids are assembled and combined together. The code lies in the sequence in one of the strands, the coding strand. Purine to pyrimidine bonding. The nitrogenous base of DNA are grouped into purines and dipurines, as you see here. And the pyrimidines are smaller as they contain a single ring structure, whereas the purines are larger as they contain a double ring structure. When bonding two strands together in a DNA helix, purines are always bonded to pyrimidines. This results in the two strands of DNA being at a consistent distance from each other. Watson and Crick discovered that the base pairings A with the T and Z with the G, with each pair held together by the hydrogen bonds. Purines are the adenine and guanine, whereas the pyrimidines are the thymine and cytosine. They are the single ring size, whereas the purines are the double ring sizes. The efficient packagings of the DNA molecules. In the nucleus, the total length of a DNA of a chromosome is over 2 meters. This length is shared by between 46 chromosomes, which are different in lengths depending on the number of the genes they contain. But we can estimate that within a typical chromosome of 5 micrometer length, there is 5 centimeter of DNA. And this means that about 50,000 micrometers of DNA are packed into 5 micrometers of the chromosomes. This phenomenal packaging is achieved by coiling the DNA double helix around protein beads called nucleosomes. The packaging protein of nucleosomes called histones are basic proteins containing a high concentration of amino acid residues with additional basic groups such as lysine and arginine. In nucleic sums, 8 histone molecules combine to make a single bead. Around each bead, the DNA double helix is wrapped in a double loop. During cell division, when the nucleus divides, the whole beaded thread is again coiled up, forming chromatin fiber. The chromatin is again coiled around a protein called non-histone chromosomal protein. The Hershey-Chase experiment. Before the middle decades of the 20th century, biologists were unsure of the genetic material of living organisms. Primarily, they debated whether genetic was based on nucleic acids or proteins. And because DNA was composed of only 4 types of nucleotides and proteins were composed of 20 types of amino acids, it made sense to many that proteins could be better provide the complex genetic variety found in the living organisms. In 1951, the Marta Chase and Alfred Hershey carried out an experiment with bacteriophage virus that parasitized a bacterium, and it helped them to confirm that the DNA is a genetic material. They made use of the radioisotopes in their experiment in a process called radioactive isotope labeling. Radioisotopes are radioactive forms of elements that can be detected within a molecule. The research grew bacteriophages in 2 different types of culture. One culture included radioactive P32 and another culture included a S35. The sulfur 35 was present in the protein code, whereas DNA doesn't include sulfur, therefore only phosphorus 32 was found in the DNA. E. coli, infected with viruses from 2 different cultures, revealed that a virus labeled with a P32 was inside the cell. As you see in the picture, the 2 bacteriophages labeled with radioisotopes were each allowed to infect the bacterium, and the E. coli infected with the phosphorus 32 labeled bacteriophage, and the radioactive detective inside the cell, a location indicating the DNA. However, the E. coli infected with the sulfur 32 labeled bacteriophage had no radioactivity inside the cell. Because DNA contains phosphorus and not sulfur, this allowed Hershey and Chase to conclude that the DNA, not protein, was the genetic material. Chargaff's rule. As mentioned earlier, scientists in the early part of the 19th century worked under the assumption that protein was responsible for the genetic traits. And research showed that there were both DNA and protein in the nucleus of the cell, and the hypothesis was that DNA existed as a tetranucleotide molecule, in other words, in repeating set units of the 4 nucleotides, and was there to help give structures to chromosomes. In the late 1940s, researchers named Irving Chargaff developed a research technique designed to show the proportions of nitrogen base types found in various sources of a DNA. His separation and identification technique involved paper chromatography. And the Chargaff's work not only showed that the proportion of adenine to thymine and guanine to cytosine are equal, but also showed that tetranucleotide theory is false. So here at the end of the chapter, you see the guiding questions revisited. How does the structure of nucleic acids allow hereditary information to be stored? In this chapter, we've described how RNA and DNA are structured. Each is composed of subunits called nucleotides. Nucleotide exists in 8 types, 4 types in RNA and 4 types in DNA. Each nucleotide contains 5 possible nitrogen base, adenine, thymine, cytosine, guanine, and uracil. In DNA, the 2 strands are held together by complementary base pairing between the nitrogenous bases. The sequence of the nucleotides in a section of DNA called genes, allowing long-term storage of the genetic code. RNA molecules are complementary copies of genes of DNA transcribed by using RNA nucleotides. And how does the structure of DNA facilitate accurate replication? So this DNA exists as a double-stranded molecule. DNA makes copies of itself. This unwinding allows the nitrogen base to make new complementary pairing using the exposed nitrogen base as a template. The pairings are adenine with thymine and cytosine with guanine. 2 DNA molecules are created from 1 during DNA replication, although neither is completely new. So the nitrogen base sizes purine with a pyrimidine and hydrogen bonding ensure accuracy when new base pairs are formed. So guys, that is the end of the chapter.